In the nucleus, we have 23 pairs of chromosomes. Each chromosome is made up of a bunch of tightly wound DNA molecules. DNA is made up of molecules called nucleotides, and each nucleotide contains a phosphate group, a sugar group, and a nitrogen base. There are 4 different types of nitrogen bases. There are adenine, thymine, cytosine, and guanine. The order of these determine the genetic code, the instruction for life.
Adenine always pairs with thymine, and cytosine always pairs with guanine. This occurs via hydrogen bonds and are represented with dotted lines in between as shown. Think of the structure of DNA like a twisted ladder. The base pairs would be the rungs, and the sides of the ladder would be the sugar phosphate. The strands of DNA run anti-parallel, or in opposite directions. The 5' end of one strand is paired with the 3' end of the other.
When DNA replicates, it first must be unwound. This is catalyzed by an enzyme called a helicase, which causes the breakage of hydrogen bonds between the base pairs. Then enzymes called DNA polymerase attach free nucleotides to the parent strands. The sugar and phosphate on the free nucleotides condense to form the backbone on new DNA molecules. This is called complementary base pairing. That means that each newly synthesized strand of DNA, called daughter strands, are made by the addition of nucleotide. That is, complementary to the parent strand. DNA replication is semi-conservative, meaning that one parent strand is always passed on to the daughter helix.
Because the DNA never leaves the nucleus, as it's too valuable to lose, its information must be copied. This process is called transcription, and the copy created is called messenger RNA, or mRNA. The first helicase unwinds part of the DNA that contains instructions to create the protein needed.
Then, the RNA polymerase pair free nucleotides with the DNA nucleotides to create the complementary copy, except uracil replaces thymine, bonding to adenine.
After transcription is finished, the mRNA leaves the nucleus and goes to the nucleus pores in the membrane. It then enters the cytoplasm to be read by the ribosome. This is when the mRNA is translated into a sequence of amino acids that make up the protein the cell needs. Each 3 bases code for one amino acid, called a triplet code. These groups are called codons. Once the mRNA attaches itself to the ribosome, it must read the start codon, AUG, to begin the translation into the protein sequence. Now, the ribosome needs the tRNA. Each tRNA molecule binds to a specific amino acid, and the other side of the tRNA contains its anticodon, which is the complementary codon for the mRNA. The tRNA is sent to the ribosome, and with their amino acids, then find their complementary codon on the mRNA. Each amino acid is bound in the ribosome, forming a polypeptide chain. The tRNA detaches from the amino acid and is sent back to find more amino acids. This procedure continues until the ribosome reaches a stop codon, where the polypeptide chain is released. The polypeptide chain will fold into the shape of the protein and continue until the protein is completed.